Hi, I'm Eric Dredden. I'm a postdoctoral fellow here at MIT's Cancer Research Institute called the Koch Institute. And I guess I'll be speaking today about some of the work that I've been doing in collaboration with Paula Hammond's laboratory in chemical engineering and also Michael Yaffe's laboratory in biology and bioengineering. So the image that we're going to be discussing today, or the one that's behind me, is really a zoomed-in picture of what is a metastatic ovarian tumor. It's quite small, it's millimeter sized, and it's actually locally invading the front abdominal wall of what in this case is a mouse model of the disease. So the color that you see in the tumor itself is indicated by a small biomolecule called an antibody that we use to label cells that have a rapid proliferation rate. And so in this particular image, this rapid proliferation manifests as in this case white color, and you can see at the bottom right-hand side of the image, you can see what is the abdominal wall. So you can imagine a mouse or a patient lying on their recumbent position. You can essentially see cells that are kind of at the single cell level that are invading the local tissue, and so of course, as a tumor, it acts as a large mass, as an ensemble, but this leading front for me is kind of like the foot soldiers that we're trying to fight back. What we know now is that the majority of ovarian cancer patients that leave the clinic with a diagnosis leave with the diagnosis of metastatic disease, either locally spread throughout the abdominal cavity or distantly spread to other sites, such as the lungs. We know that these metastatic tumors are more difficult to treat because they are very invasive, and they also have some unique mechanisms, biochemical mechanisms, by which they decrease their sensitivity to chemotherapeutics that are used to treat tumors today. In our case, we're particularly interested in decreasing proteins that contribute to the recognition and repair of damage caused by traditional chemotherapy. And so our treatment strategy here really relies on a technique called RNA interference. RNAi is, in my view, incredibly powerful, in part because what it allows you to do is to deliver a nucleic acid strand that can essentially decrease the expression of any disease-causing protein. And so our hypothesis is that by decreasing the expression of these chemo-resistance causing proteins, that we can sensitize these metastatic ovarian tumors to therapy that's being already used in the clinic today. And so the challenge for RNAi is that it turns out it's incredibly difficult to get nucleic acids to circulate for long periods of time. They're very small. They're very susceptible to degradation in the blood, and it's really tricky to get them inside of cells and also into subcellular compartments at which RNA interference occurs. So what we do is to design polymers that help us to package these nucleic acids into structures that are large enough to circulate, that can shield them from biomolecules that would normally degrade these nucleic acids in circulation, and also help them to get inside and also localize in subcellular compartments within cells at which we can achieve RNA interference. So what we're seeing is that in vitro, in cellular models, and also in higher mammal models, that we see improved sensitivity to frontline chemotherapy, and we also see improved treatment response in terms of tumor volume, and also, of course, most importantly, improved survival as well.
Anything else about the image itself?
I like the colors.